it's a beautiful segue because decision making in this particular space is especially vexing. Really, it's exacerbated. Like we're not making monoclonal antibodies here. Like we're working in science that is changing all the time. Influences are changing all the time. So let's start talking about that a little bit. As you guys formed up this, scientific concept and translated it to a business. Obviously, you don't do that without paying attention to what's going on around you in this particular space. So how have your perspectives on the RNA space at large, on where it's going, what it needs?
I'd be really curious to hear just, your sort of take on where the space has come from, right? And where you see it going broadly, kind of removed from your company at this moment. Like where are some of the big trends you're seeing that space moving towards? And how that's, what are you most excited about, or what are you most perhaps anxious about?
That's not what I was going to ask, but you hijacked my question.
I told you I can't be controlled when you put a mic in front of me.
I think we just asked Sophia and Eerik like 7 questions in one.
That's how I do it. So pick one.
The great thing is pick your favorite one.
Yeah.
Pick your favorite one.
Mm-hmm. Yeah, mRNA has always been exciting. Like I mentioned, Moderna success has been a long time coming. But as I alluded to before, if you just have this print statement, you're just making that protein from that mRNA therapeutic, you might be limited to just some. I'm going to say the low-hanging fruit. Of course, it's a lot of effort to make all these medicines, but in terms of the thinking around what you're making and how your application space is limited. So you asked us to detangle a little bit from the company, but I do think the most exciting part is being able to say where and when we're making these genetic medicines, ultimately the proteins that affect the function that are encoded in these mRNAs. So having looked at and now lived at the mRNA space a bit more in more detail, big challenges are still delivery, but what's exciting there has been maybe a shift in the business model a little bit. We're seeing a lot of companies who are innovating on the delivery space who don't necessarily have the ambition to make therapeutics themselves. They want to work with other folks who have other innovations on the mRNA molecule or the disease to go after, and they can provide the vehicle to get there. So we're kind of seeing a little bit of progress there in now having the thing you need to encapsulate your stuff in, right? And that's very important versus having to now build an RNA company and a delivery company at the same time. And that's what we've maybe seen before, where there's a lot of mergers where there's a delivery company and an RNA therapeutics company, and they merge to have more efficiencies. But we really see that the delivery vehicles could be reused in other applications by other people, and nobody has the bandwidth to do everything. So I think it's exciting to see that this is becoming a bit more usable by other folks from that perspective. At the same time, mRNA has been around for a while, so there's certain innovations that are coming off patent, and I think that can invigorate some innovation or using of these components as well, and I think that's exciting. mRNA is a platform, and these delivery vehicles, they all are platforms, and that has benefits and has challenges as well. The FDA has been thinking about the platform regulation quite a bit, and I think there are many nuances to really think through there. For example, if you think about an mRNA therapeutic, there's kind of 3 platforms there typically. There's the delivery platform, there's the payload platform, the mRNA molecule, and then there's the thing you're encoding. Say that it's a genome editor or something like that. Each of those is a platform on its own and could be de-risked in different ways. So how do we, in the future, I would like to see how do we think about all of these components coming together and really efficiently de-risking all of these components and on the regulatory side so that we actually can make the medicines quicker. And the promise of these platforms is that we can get to patients quicker so that they can benefit versus having to start from scratch again every time we're making a new therapeutic, even though we change just one aspect or brought together these different components. So that to me is exciting. I am still a molecular thinker, in my heart. So there's just so much interesting molecular biology that's happening, being either invented or discovered. We are, of course, on the front lines there, having used existing biology of this within cells, but also discovered new biology, which has just been exciting because it really can make a difference to patients that currently don't have any treatment options. That's my take.
Yeah. I was going to say, Sophia, too, you can kind of build on that as well in terms of how are you seeing some of these evolutions that Eerik's excited about, that are exciting you, right? That are kind of shaping how you anticipate you would like to shape your own strategy, right? As you're entering the space as a brand new company and trying to carve your own niche, right?
I'll say a few of the same things Eerik said in maybe a different way. The way that we see the delivery space evolving is right now, many companies are solving either one or two questions. It is biasing these vectors towards new tissues so that they don't all just sink in the liver, and or targeting as well. So I think it's understood if we want to move towards in vivo programming or in vivo use of genetic medicines, we need to get it into the right tissues, and we need it to get into the right cells or really only be active in the right cells. So there's biasing and there's specificity. And I think that there's a lot of progress on the biasing front. And I think specificity by modulating just the vector has been very hard. For many reasons. We have an op-ed on Cell and Gene, so go read it if you want to read on
That's
why
a great plug.
Yes, I know it's related. So,
yep.
Yeah, so for many reasons has been very hard. And so we at Radar think about this in a whole different way. We think we want to read the universal code of the cell. And we really believe the RNA profile is the universal code. It is the information that tells you how static genes, so static DNA, that's the same in all cells, is translated into the dynamic protein profiles that are really driving individual cell behavior. So if you can read this RNA profile, the AUCGs that make it up, you can really be reading that universal code that tells you what the cell is doing. Why don't we use that code to find specificity? Why don't we use that code to tell us exactly what cell we're in or what that cell is doing that makes it our target of interest? And so we like to think about it a whole different way. Instead of making your vector find a cell surface marker that potentially defines a cell type, might be shared by others. Let's think about it, just reading the code. Let's read the code straight. And so it's very clear the entire field is thinking about this. It's thinking about in vivo, going in vivo. That is the way that we're going to make these genetic medicines scalable. That's the way that we're going to make them cost effective. That's the way that you're going to be able to take them from just being able to be delivered at very specialized academic centers to clinics that can do, just IV transfusions or subcutaneous injection to modes of administration that can be available to more patients. It's the way you're going to drive down cost. Everyone is thinking about taking things in vivo. It's the next frontier. To do that, you need to have exquisite specificity, particularly to get at very complex, high burden indications that affect broad populations. And that's why we at Radar, and I want to say, when you found a business, you can take your values and make them into strategy. And our strategy really is to make medicines for all people, as many people as possible. We have to have design criteria that say, we're going to innovate in a way that makes a step change in safety and a step change in accessibility. And that means we have to drive down costs. We have to do this in vivo. We have to be able to target diseases that are affecting broader populations. And I'll say this is a shift. I think about 5-6 years ago, I was in China actually still, and I remember giving a presentation on the promise of CRISPR-Cas9. But at the end of that presentation on the technological promise, there was still an understanding that this would be a technological feat to most patients affected by diseases they were going after. And, I think Casgevy it's a huge success that we have Casgevy approved now for sickle cell disease. But sickle cell affects African Americans at a higher rate. Are they going to be able to access a medicine that it's, on average $2.1 million or more that has to be delivered at an academic center where you have to have your cells removed from the body, isolated, re-engineered, put back into the body 30 days later, and then potentially have time in the ICU? Oftentimes the hospitalization cost is half the cost of that treatment. And that's just not going to be accessible for most people. It elucidates a lot of biology. It makes us know that it could be possible to move forward. But I think everyone knows we have to bring it in vivo. Same for the very promising CAR T therapies. Also, when I was in China, I wasn't always in China, but it just happens that a lot. I was on a panel, through the National Committee on U.S.-China Relations, there was a panel between Chinese regulators and U.S. regulators talking about CAR T, the promise of CAR T. I think there was almost a competition between the U.S. and China at the time, accelerating the CAR T trials. But even then, we were talking about this has to be ex vivo today. So these are the kinds of patients we can serve, the very, with very severe cancers. There was, this idea that maybe one day it could be in vivo. It was just an idea. Now it's a big idea. Now people are trying to make it happen. And so in vivo is definitely the future. That's what we believe at Radar. And we believe we have a very, now I'm moving to the pitch to Radar, but I do think that's the future. The pitch to Radar is that we really think that you need, if you want to get that exquisite specificity, read the entire code of the cell. Read the entire code of the cell and do it in a way that you can make it accessible to people. So think about the cost think about the route of administration. The last part I'll say is that what's also exciting is there's always been this idea, platform versus single asset, and platform. And what is a platform technology? To be a platform technology, you have to be able to develop multiple products from a similar concept. And I think, at the end of the day, what matters to patients, what matters to pharma, what matters to VCs is the eventual quality of the product you generate. And the promise of the platform is that you could generate one product, then multiple products after that. And I think with Moderna, we have these first products, the COVID vaccine, the RSV vaccine, and the promise of more. You're starting to actually see platforms fulfill the platform promise, which is high quality products staged. That's also what we think is very promising at Radar. We want to form high quality products. We really believe that if we get it right the first time, those wins will translate to more medicines. So I still think that there's a lot of promise in platform as long as we make high quality products.
Well, I asked the last question, and you said I asked the wrong question apparently. So we'll go.
Once again, I'm offended.
It's your turn, Matt. Go ahead.
We're gonna run short on time. I can tell you that.
I know.
We're gonna have to motor, and then we'll have to do a part 2 or so.
I know. You're just gonna have to come back.
So I could go one of two ways with this. The platform conversation that you just started, Sophia. I'm curious about, and this is sort of maybe skipping to the end. Where on my normal story arc on the business of biotech might be skipping to the end. We'll backpedal if we need to. But can you share any early thoughts on what a potential pipeline might look like for Radar Therapeutics?
Yeah.
And I don't mean to paint you into a corner or ask you to say anything that you're not, your board is going to be upset with you for saying, but to the degree that you can share.
Yeah. For us, our pipeline, we really think about where can we create a step change in safety and a step change in accessibility, and where do we have the potential to be first in class? So these are guiding for us. We are looking at very complex high burden diseases. So the logic has to be these cell types cannot really be isolated in vivo or targeted in vivo without an element like Radar, without an element that really gets to the universal code and then selectively activates that mRNA. Which are the cell types that are currently inaccessible by other technologies or not able to be truly specifically targeted without a technology like Radar. Those are our guiding principles. We have identified a few areas as very promising. I think I won't mention them, but if you dug in to certain public places, you can educated guess at what we're really excited about.
Mm-hmm. But I'll say in each of these systems, there is always a pipeline behind it because you have this very complex cellular environment. Once you make a sensor or a control element for a particular cell type within it, you have a pipeline of products that could come after it. Because in these complex environments within the body, many things can go wrong. You have a complex engine, and any of those parts breaks down, you get a disease state. So that's how we thought about it, that there's kind of a franchise, a pipeline behind each of these efforts that where we can reuse the components, reuse the learnings, reuse the expertise and capabilities we've built out, and really fulfill that platform promise. So that's always been kind of a principle as well.
Do you anticipate that will down the road ease the regulatory rigors as well, Eerik? I mean, is that sort of something you're thinking about early on?
We hope so, yeah. And we are always gathering insights from our advisors, from their experiences, particularly in the nucleic acid therapeutic space, where sometimes, say, a mouse model might not make sense because you're dealing with sequence interactions, and you really want to deal with the human versions of these transcripts you're interacting with, or other components within the cell. So we're looking to see how have other companies addressed this challenge, maybe working really heavily in primary cells, and then maybe doing an NHP safety study, and to prove to the regulators that this is safe. But then convincing yourself on the efficacy side, mostly with primary cell data, looking at actually donors, patient cells, and seeing how your mechanisms work there. So by working in similar systems, it can help us accumulate this data on safety and performance. And I hope that this can lead to acceleration in the path to approval